All right, honors biology. This is Unit 2 biochemistry, and this is going to be Part 3, making and breaking polymers. Make sure you write that title down. All right, so let's start talking about dehydration synthesis. Remember, when you get dehydrated, it's when you're kind of removing water from your body. So think of that when you think of this dehydration synthesis. Dehydration synthesis or condensation, they are synonyms for each other. It's when we build polymers out of monomers. So we take those little teeny tiny parts, those little individual portions, and we take water away from them and we build polymers. Water is released and monomers are dehydrated. If we've already done it in class, remember the little demonstration? Maybe I had you guys come up in front of the class and you guys had an OH in one hand and an H in the other hand. Remember, water is H2O. And when I put that water into your hand, you guys were monomers. And when I dehydrated you or took it apart or took it out of your hands, you guys could connect to build a polymer. So that's dehydration synthesis. Okay, an example of that is how we form a disaccharide. Remember, di means two, and then saccharide means a simple sugar. When we put more than one monosaccharide together, like glucose, so here's my monomers down here, okay? When I put those monomers together, or I dehydrate them, taking this water away, I end up with a disaccharide, okay, or two sugars. So monomers, polymers, okay? Show you here. So we have the OH group on one of my monosaccharides, okay? So over here are my monomers. Whoop, there's my OH. And then the H on the other one, okay? So we're going to dehydrate. We're going to take this H2O out, okay? We're going to take that H2 out. And if you can see over here, all that's left is the oxygen. Then over here in my disaccharide, when I connect them together, that O connects my two monomers together to make a polymer. All right. So when I take those things out, it leaves my two monosaccharides joined by this one oxygen. And remember, when I put two monosaccharides together, I get a disaccharide. Okay. So these are monomers right here. Monomer, whoop, monomer. Remember, glucose is a monomer. And then when I put the two together, I get maltose, which is a polymer. And it's an example of a disaccharide. All right, so I just dehydrated it, took out this H2O. So I end up with two monomers together, and then you can see right here, here's my H2O. I end up with the water off to the side. Woo! Alrighty, friends, now here's an example of dehydration synthesis with proteins. We already kind of did it with carbohydrates there. I showed you that disaccharide. So now here's an example with proteins. You guys can see you've got two amino acids, and those amino acids come together with a peptide bond right here. So these two amino acids come together, peptide bond in the middle, and then water comes out. Okay, so just another quick example where we have dehydration synthesis. Water's coming out, and they're synthesizing together to make one macromolecule. All right, now hydrolysis, the opposite. Hydrolysis takes polymers and breaks them down into individual monomers. It's going to put water into those monomers. So it's going to take, once it breaks the polymer apart, it's going to put that OH and that H back onto your monomers to break them into, back onto your monomers to break the polymer up. So think when you hydrate, you put water in. Hydro means water. Okay, an example would be breaking starches down into their individual monosaccharides, okay? So here, this is dehydration synthesis. We already talked about this one. When we have individual monomers, we put the water in. There's my H2O right there. And then it helps to connect them. We take the water out. We dehydrate, sorry. We take the water out right here, H2O, and then we end up with one big polymer plus water. Okay, so that's dehydration synthesis. Now focus on breaking them apart. Okay, let's focus on putting water in. All right, so here is my big long polymer right here. Woohoo! All right, big long polymer right here, all connected together in one. They're all holding hands. And they're all connected with an oxygen in the middle. Now what we want to do is we want to add water to that and enzymes, which help to speed up chemical reactions, remember. So the enzyme is going to help to break apart the polymers, and then we can go in and add our H2O and end up with monomers. Okay? So and we break them up. Now we've got our individual monomers. We hydrated. We filled their hands with the water again, just like we did that example in class. Sorry guys, I went back a few too many there. Okay, here we go, the rest of hydrolysis. So here you guys, this is dehydration synthesis again. The bottom is the hydrolysis picture. So your enzymes split your sugar at the oxygen. Then we're going to add the water in, H2O, to fill in sort of the edges of our monomers. They're going to be kind of an open site. And then the polymers get separated into monomers. And that's hydrolysis. Remember, you're saying dehydration makes, hydrolysis breaks. Dehydration makes polymers, hydrolysis breaks polymers. Quick review over the last 2 videos so far. Make sure you know what are the monomers and polymers of each of the 4 macromolecules. What are the functions of each of them? What happens during dehydration synthesis? And what occurs during hydrolysis. So make sure after videos 2 and video 3 you can answer all those questions. And that's it for video 3, unit 2, biochemistry. Woohoo! Good luck, my friends.